In Activity 10, Birth and Growth, students observe and describe the birth and growth of snails and platys. They examine a cluster of snail eggs and observe the development of the eggs over time. They learn that some fish lay eggs, while others give birth to fully developed young, and finally compare their own birth and growth with that of snails and platys. You will need the following materials from the kit: Activity Sheet 10, Parts A and B, Aquariums from Activity 9, cover slips, depression slides, plastic dishes, droppers, magnifiers, plastic spoons, toothpicks, dip nets, fish food, grease pencils, pocket scopes, and masking tape. You will also need to provide crayons, paper towels, pictures of different types of eggs, and spring water. To prepare for Session 1, make a copy of Activity Sheet 10, Part A for each student. The presence of snail eggs is required for this session. When you see eggs on the sides of the aquarium, draw a circle around them in grease pencil on the outside of the tank. If snail eggs are not yet visible, you may want to go to Session 2. Start Session 1 when eggs appear. Also, collect pictures of different types of eggs for students to view. For this session, each team will need their aquarium if it contains snail eggs, a plastic spoon, a plastic dish, 2 magnifiers, 2 sheets of white paper, some crayons, and a small piece of tape. They will also need some spring water. To begin Session 1, ask students if they have noticed small fish and small snails in the aquariums, and elicit that the platys and the snails have had babies. Distribute pictures of different types of eggs and challenge students to guess what they are and where they came from. Then explain that an egg is the beginning of a new baby animal. Next, have the teams retrieve their aquariums. Distribute 2 magnifiers and 2 sheets of white paper to each team. Have the students place their aquariums on a sheet of paper for better viewing. If there are snail eggs present, help them identify them. Some students may think they're a type of algae, while others may have noticed a similarity to some of the eggs in the pictures. Then, instruct students to gently transfer the jelly-like mass of eggs from the aquarium to a plastic dish with a small amount of spring water and a small sprig of anacarus. Inform students that snail eggs take between 2 and 3 weeks to hatch. Next, distribute a copy of Activity Sheet 10, Part A to each student and assorted crayons to each team. Have students draw a picture of the snail eggs in the first square labeled Day 1. Explain to the students that they are going to observe the eggs every other day for the next 3 weeks and record any changes they see. To conclude Session 1, have students return their aquariums and dishes of snail eggs to the storage area. Return the other materials to the kit. To prepare for Session 2, make a copy of Activity Sheet 10, Part B for each student. The presence of young fish, called fry, is required for this session. When you see any fry, promptly move them to their own tank so that they will not be eaten by the adult fish. Add a small snail and a few sprigs of anacarus to the nursery. Place the nursery, a dip net or plastic dish, a jug of spring water, and a container of fish food at a distribution station. For this session, each team of 4 will need 2 magnifiers, some crayons, 1 sheet of white paper, a plastic dish, and a pinch of fish food. To begin Session 2, bring students' attention to the baby platys in the nursery and ask, where did these tiny fish come from? Explain that some types of fish lay eggs, while others, like the platys, give birth to babies that are fully developed. Have the teams retrieve their aquariums. Distribute a copy of Activity Sheet 10, Part B to each student and the materials to each team. Tell students to bring their dish to the distribution station and place a platy fry in each team's plastic dish. Inform students that baby fish are called fry. Have students sprinkle half of the fish food in the aquarium and grind up the other half into tiny particles and sprinkle it into the fry's dish. Then ask students, how are the fry like the adult fish? Explain that like the adults, the fry have 2 eyes, fins, gills, and can swim. They also eat the same food that the adults eat and can find it on their own. Then ask, how are the fry different from the adult fish? Students should observe that the fry are much smaller and less colorful. In fact, students may be able to see the bone structure and internal organs of the platy fry through the fish's translucent skin. Guide students to color in the picture of the adult platy on Part B of the activity sheet and add drawings of the platy fry next to the picture. Next, ask students to note where the platyfry are hidden within the anacarus leaves. Explain that the fry hide in plants to avoid being eaten by other animals, including adult platys. Finally, tell students to check their tanks every morning for new fry. If they see them, they should bring them to your attention so that they can be moved to the nursery. To conclude Session 2, have students return the fry to the nursery and place their aquariums back in their storage place. Collect the other materials and return them to the kit. To prepare for Session 3, send a note home to parents asking for students to bring in a baby picture of themselves as well as a more recent photo. Students may instead cut out pictures of baby and adult animals. For this session, each student will need his or her copy of Activity Sheet 10, Part A. Each team of 4 will need their dish of baby snails, pocket scopes, 2 magnifiers, 1 depression slide, 1 cover slip, 1 dropper, 1 toothpick, 2 sheets of white paper, photographs of themselves or pictures of baby animals, and some crayons. To begin Session 3, ask students to retrieve their dish of snails and their copy of Activity Sheet 10, Part A, which shows the growth and development of their snail eggs over the past 3 weeks. Then ask, what did you notice about the eggs that first week? The student should have noticed a small, brown spot appear in each sphere. The spot grew larger over time, then disappeared. Ask students, what do you think happened to the dark spot in the eggs? Have them observe the dish carefully. If necessary, point out the small snails in their dishes. Lead them to conclude that the dark spots were baby snails inside the eggs. Point out that many animals go through changes or different stages as they grow from birth to adult. These changes are called the life cycle. Distribute a depression slide, a cover slip, a dropper, and a toothpick to each team. Instruct students to pick up a baby snail with a toothpick and place it in the well of a depression slide, along with a couple of drops of water from the dish. Have them cover the slide with a cover slip and view the baby snail under the pocket scope. Students should see that baby snails look just like adult snails, except smaller. Have students add a drawing of this baby snail to the last square on their activity sheet. Then ask, are baby platys born the same way baby snails are born? Students should understand that platys are born fully developed, and snails hatch from eggs. Finally, ask students to share with the class their baby pictures, or the pictures of baby and adult animals they cut out from magazines. As a class, discuss how newborns are very small, but grow to be roughly the same size as their parents. To conclude this activity, you may not want to add the baby snails to the aquariums. See Materials Management, page 13, for suggestions on how to handle organisms you no longer need. Discard the toothpicks. Collect the dishes, slides, cover slips, and droppers. Rinse and return them along with the magnifiers and pocket scopes to the kit. For science background, reinforcement activities, curriculum connections, and information about the Delta Science Reader, please consult your DSM Teacher's Guide.